Hello, everyone. Welcome to today's live broadcast, Introduction to Exosomes and Other Extracellular Vesicles, the body's molecular internet for personalized and precision medicine. I am Brenda Kelly Kim of LabRoots, and I'll be your moderator for today's event. LabRoots.com is the leading scientific social networking site, and we are proud to bring you this interactive web seminar. For more information, please visit us at www.labroots.com. Here's how this presentation works. We want to hear from you. Questions, comments, and even answers can be submitted via the green Q&A button in the lower left of your screen. We'll try to get to everyone, but if not, we'll definitely make sure we follow up with you by email. You can enlarge your slide window by clicking on the screen icon in the lower right-hand corner of the slide window. If you can't hear or see this presentation properly, definitely let us know by clicking on the support button at the top right or the Q&A button lower left. This is an educational web seminar and so offers free continuing education credits. After the webinar is over, please click on the CE button located in the bottom left-hand corner of your webpage and follow the instructions to claim your credits. I would now like to introduce today's speaker, Rey Magana. Rey Magana is a serial entrepreneur with over 25 years experience in life sciences. Mr. Magana is currently co-founder of the Exo Incubator, a life sciences technology incubator, and Accelerator Exo Incubator's mandate is to exploit the power of its exosome and extracellular vesicles platform technology to potentiate precision medicine and address critical areas of an unmet medical need. Among the disease areas of interest are oncology, metabolic disease, genetics, immunology, and infectious disease. Previously, he was part of the team that pioneered extracellular vesicle and exosome nucleic acid technologies for applications in molecular diagnostics, drug delivery, and therapeutics. He co-founded Proxy Life Science Holdings, a biotechnology company organized to develop those technologies. He also co-founded Perry Scientific, a contract research organization specializing in preclinical research services supporting early-stage pharmaceutical, biopharmaceutical, and medical device companies. I will now turn it over to Rey for his presentation.
For the introduction, Karen. I also want to thank LabRoots for hosting this talk. Good afternoon and good evening to all attendees. Thank you for joining us. My talk is titled The Introduction to Exosomes and Extracellular Vesicles, Implications for Personalized and Precision Medicine. Before we begin the talk, I do want to share a disclaimer that I am a co-founder and stockholder in Exo Incubator, and therefore I have an interest in various technologies, companies, and patent pools, which include the following two patents, which are in the field of exosomes, extracellular vesicles, and nucleic acids. And with that, we'll get started. The discussion, I've basically broken it down into four main segments. The first will be a brief history of exosomes and extracellular vesicles from their origins to our current understanding. The second section will involve, it will be on the involvement in physiology and pathology with a focus on human disease. And the third, I want to discuss why I believe there is a fertile environment for this technology at this time and its potential role in personalized medicine. And finally, the fourth segment will focus on practical applications, and we will talk about two important areas, their use in research and development as a platform, and also their use for development of novel medical diagnostics. So the first section will start with what are extracellular vesicles, and we'll cover a brief history of their origins and our current understanding of their functions. And so extracellular vesicles and exosomes comprise a newly discovered communication system, which we like to call the Internet of the Body. And it's a very fundamental system that affects every cell and organ, and it's believed that every cell type produces them, and they are involved in both normal physiology and pathology, and they circulate in biofluids. And each exosome or extracellular vesicle is representative of the cell from which it originates, and we'd like to call them proxies for those cells. Now, they comprise an extensive network which interconnects various biological systems. And so by collecting these exosomes and extracellular vesicles and interrogating their content, we essentially have a very powerful tool that we can use to gain really unprecedented insight into the molecular activity and the crosstalk that is taking place. In the future, this may be done in real time, and currently it's non-invasive since it can be obtained from biological fluids. The vesicles, extracellular vesicles are derived from membrane-bound structures. They're released by cells into the extracellular space. And there are essentially two main categories. Exosomes are released from cells when endocytic multivesicular bodies fuse with the plasma membrane, whereas microvesicles are released directly from the plasma membrane via blooding. And here's a nice example of platelet microvesicles being released as an electron microscopy image. Just to give an idea of relative size, the exosomes, which are the smaller versions of vesicles, are in the range of viral particles, whereas microvesicles are a bit larger and they're in the range of bacteria. Apoptotic bodies, which also fall in the definition of vesicles, are a bit larger. And although, again, they are included in that definition, for the most part, when referring to extracellular vesicles, they're mostly referring to exosomes and microvesicles. We try to differentiate that by specifically using the terminology for one or the other, but since the beginning, they've been used interchangeably. And so the reason that exosomes and microvesicles are an exciting entities to study, and they have a lot of potential for applied research and translational research, and eventually in the development of diagnostics and even therapeutics, is that they are a rich source of information. And they derive that information because they carry macromolecular cargos in the form of proteins, nucleic acids, bioactive lipids, as well as metabolites. It's estimated that about 3 million of these little critters can be found in per microliter of serum, and they represent a heterogeneous population. So because these exosomes and microvesicles are produced from various different cell types, they are represented in the circulation. Of note, these vesicles are of most interest because they are functional and they are bioactive. Indeed, one of the very first experiments that led to that notion was that people observed that these vesicles were elevated in cancer patients as compared to in normal patients. The other important I think property is that these are relational data sets in a way, because they all derive within a single exosome. So you can find these macromolecules in a single exosome, whereas other circulating proteins or nucleic acids are usually independent, and you don't see that relational aspect to it. And so these vesicles, because of the surface, they carry a lot of surface markers that are associated with the cells from which they originated from. They can be identified in terms of their tissue specific sourcing. And part of the reason that they are bioactive is that they are rich in their cargos. And for instance, they carry many types of membrane proteins, including oncogenes and growth factor receptors, integrin receptors, MHC class 1 molecules, soluble proteins such as proteases and cytokines, as well as signaling proteins and transcriptional regulators, reverse transcriptase, and transmembrane proteins. Indeed, out of the 4,400 proteins that have been identified, and that number is probably much higher now, many of them are molecular markers that are associated with cancer. For instance, those listed here: VEGF, HER2, EGFR, and EpCAM. And again, these markers can also and have been used, and work has been published where you can pull specific populations of exosomes using these markers. For instance, using immuno beads. Nucleic acids have also been found, including DNA and RNA. The DNA, there's both genomic and complementary RNA that's been identified. And as far as RNA, there's over 1,300 different molecules, both coding and regulatory. Exocarta is a very good source. It's a comprehensive database of the various proteins and nucleic acid contents that are found in exosomes and extracellular vesicles. With regard to RNA, there's messenger RNA, non-coding RNA, and microRNA, among other species that can be found within them. One of the first reports of these extracellular vesicles was back in 1967 by Peter Wolf. And in his case, he observed them in human plasma as he was studying platelets and coagulation. And at the time, he called them platelet dust. And also, they were known early on as microparticles because they were not well characterized at the time. And you could see, actually, he also was the first to identify that you could isolate these using ultrasonification and that they were rich in phospholipids. Most importantly, one of his findings was that they showed coagulant properties resembling those of platelet factor 3. And so platelet factor 3 initiates the blood coagulation cascade. And he noticed this activity, and just to test it to see if it was some other factor in the plasma or these particles, he removed the particles and then saw that the activity was no longer there. So he showed in these experiments that the bioactivity was associated with these particles themselves. Now, despite the fact that this is very interesting results and observations, not much was done after 1967 until 1983, when work was being done at Dr. Stahl's lab by Harding and others, that studying the transferrin cycle. And so the transferrin cycle is research looking at the recycling of iron-binding blood plasma glycoproteins using reticulocytes and immature blood cells, looking at the iron receptor complex, wherein these complexes form endosomal membranes at the cell, at the plasma membrane, and are internalized and are converted to multivesicular bodies, which are essentially exosomes, or what eventually become exosomes. And so they are recycled back out, releasing their contents into the extracellular space as exosomes. And here you can see from their work a couple of micrographs where you see the multivesicular bodies. And you see also their fusion and release with plasma membrane, where ultimately here you see a vesicle on the surface of the reticulocyte. And these are labeled with gold particles, and so that's how you're able to observe them. So this is one of the, if not the very first observation of the mechanism by which they are exteriorized. And so a brief timeline, which includes a little bit of what we already spoke of. So in 1957, apparently was a period when multivesicular bodies were being studied. But at this time, I don't think anybody knew or even postulated that they were exteriorized, at least not as functional entities, other than maybe in apoptotic bodies. In 1967, that's Wolf's work, where he identified the platelet test. And then in 1983, there's the Harding and Stahl work with the multivesicular bodies. And in 1987, Rose Johnson, which also was working in 1983 and so on multivesicular bodies, she's the one that is credited with giving or coming up with the exosome terminology. A very important paper was published in 1987 by Grasso and others, where she was working with antigen-presenting cells, and she found that dendritic cells produced exosomes, and that these exosomes expressed MHC1 and MHC2 molecules, and that they induced and enhanced antigen-specific T-cell response in vivo. So this was one of those seminal papers. And in 2002, an industry group comprising these two individuals, Dr. Acher and Dr. Lepek, were the first, as far as I know, to create recombinant exosomes and to use them and postulate their use for as immune modulators, as therapeutics, among many other uses for them, including drug delivery. In 2003 and 2005, again, Dr. Johnston, working now more fully in the field, decided to promote and establish the first meeting on exosomes. And around that same period, this is when I got involved in the field with a group led by Dr. Kruskovsky in 2004 and 2005. And our group was the first to identify exosomes and extracellular vesicle RNA and DNA cargos, and also to identify coding and regulatory RNA and their horizontal transfer. In 2006, an academic group led by Dr. Radicek confirmed these findings and also showed reprogramming using RNA horizontal transfer. And then in 2007, Dr. Velati and Dr. Lotvo also confirmed these prior results and extended the known number of entities that the exosomes carried, and so they created a very large list of mRNA and miRNA sequences that they found in exosomes and extracellular vesicles, and they also confirmed the RNA transfer. And in 2008, Skog and Breakfield out of Boston also confirmed that work further in disease glioblastoma, and they also identified and confirmed the RNA transfer associated with glioblastoma. And then very exciting to me, because I come from the preclinical field, they in 2012, Lou and Dr. Wood out of Oxford were able to produce recombinant exosomes and show delivery in vivo. So they injected IV these recombinant exosomes that were loaded with various markers and RNAi type therapeutics, and they were able to show delivery to specific brain cells across the blood-brain barrier, which I think is pretty significant. So since 2010, we see an exponential growth of interest in the field and of academic papers being published. Over 1,700 papers were published during those last 3 to 5 years. And also the International Society for Extracellular Vesicles was formed, and there's over 700 scientists and academics involved. So most of this interest was from academia, but more and more nowadays there's interest from other groups, including commercial and industry groups. By the way, Select Bio has published several reports. They've been following this field very closely for a while. And if you're interested in looking at the various publications broken down in a lot of detail as to the specific biomarkers that they were looking at, or whether they were looking at exosomes or microvesicles, or their applications, whether they were looking at RNA or protein content, these reports break it down very closely, and they also have other reports on the field in general. So I encourage you to look them up. They're available online. Very interestingly, the National Institutes of Health have taken an interest in this field as well, and they have coined the term xRNA to describe it, yet another term. They also designated a common fund project, and my understanding that this is a sort of a privileged designation, and is reserved for very special circumstances and projects. And so I think they do a very good job of sort of representing how special this field is. In order to qualify for this designation, it has to be a unique area of research that has transformative potential, that is very catalytic, and that is synergistic with other areas of research and other technologies, and is cross-cutting. And indeed, I think if we don't all agree yet, perhaps after a few more slides here, we might all agree that certainly exosomes and extracellular vesicles fall within that definition. So we'll go now a little bit to their physiology and involvement in pathology, particularly in human disease. And so they are, interestingly, very conserved across very divergent species. And so I think this is a sign that they are essentially a very fundamental phenomenon. And of course, most of the work is done in mammalian models nowadays, but they've been identified in bacteria and in plants and in parasites. And in developmental biology, they've been identified and have been referred to as morphogens for quite some time. And so this is interesting because some of our early work was with stem cells, so it makes sense that they had been looked at. But again, the extracellular vesicles is terminology that's fairly recent, but they've been known as many different things, including as referred to earlier as dust particles, microparticles, microvesicles when I got involved, and then as exosomes as a subset, and they're also referred to by their tissue of origin. So there's prostosomes, and then there's dexosomes for those that are derived from dendritic cells, and many other names, including oncosomes. Interestingly, they're found in all major biological fluids. As far as I know, I don't know that there's been anywhere they haven't been found. And so it includes blood fractions, serum and plasma, and urine, saliva, cerebrospinal fluid, even in breast milk, and ascites and synovial fluids, even the semen, and sweat and tears. And there were some talks yesterday that covered their saliva research. So in pathology, they've been implicated in a myriad of diseases. Those include diabetes, cardiovascular disease, and many neurological diseases, including Alzheimer's, Parkinson's, multiple sclerosis, and also in various autoimmune conditions like rheumatoid arthritis and lupus. They've also, in prenatal research, they've been, DNA and RNA has been identified in microvesicles that come from embryonic maternal blood. And so these could have potential applications in fetal genetic mutation analysis. I know there's been some publications that even sexed embryos using exosomes, and they've been used in research in preeclampsia. In oncology, hematology is a big area where they've been identified and are involved in various pathogenic events. They are also being used by a group to develop diagnostics, and it's expected that those tests might be released in the next year or so. They also have been involved in almost every solid cancer type, including those listed here, and in metastatic lesions as well, which is really important when you're talking about trying to understand and prevent or reduce mortality in patients, as metastasis is the biggest source of that. Of fundamental importance is their horizontal transfer of their contents, and of most interest for us is it's their RNA transfer. And so these molecules act as a shuttle system, delivering bioactive molecules, whether it's by ligand receptor interactions or taken up by endocytosis or via fusion with plasma membranes. The transfer, it can induce transformation of the recipient cell, and they certainly affect phenotype, and they affect growth, division, differentiation, stress response, survival, apoptosis, and many, many other things. And a lot of these functions are published works in those 17+100 publications that are out there. And so I won't go into a lot of the activities that have been published related to this, but because we're going to be a little bit short on time and a lot to cover, but certainly I'd be happy to send you some references and links if you're interested. There's a bit of a dichotomy in that they're involved in normal physiology, and also in disease. And so with stem cells, they can modulate phenotype, and as I mentioned earlier, they've been identified as morphogens in developmental biology. In immune response, they carry inflammatory molecules and are involved in antigen presentation. And in the nervous system, they carry neuroprotective factors, and they act as trophic support for axons. And then on the red side of things, in cancer, as mentioned earlier, they are involved in growth, invasion, and metastasis. And in neurodegeneration, they can spread pathologic characteristics to other regions of the brain, and that is seen in cancer as well. Again, they're involved in many different disease mechanisms, and in particular here, there's examples of their involvement in cancer. And so these are activities or functions that they found in tumor-derived microvesicles. And some of them I'll go through them very quickly, but it's modulation of the microenvironment to support tumor growth, horizontal transfer of VEGF and mRNAs and promotion of angiogenesis, evasion of immune surveillance, cloaking of tumor cells from the immune system. In invasion and metastasis, they carry protease cargoes that facilitate matrix degeneration. And also in the transfer of aggressive phenotypes to horizontal transfer of oncogenic receptors and RNA. Multi-drug resistance through efflux from cells. And again, there's a lot more information that could be discussed, but if you're interested, let me know, and I'll send you some links and some references. They're also very involved in immune function, in particular in those related to tumorigenesis. And so the slides here, I'll just read off from the notes here, but it's basically exosomes released from tumor cells affect the focal tumor environment, remodeling extracellular matrix and promoting vascular genesis and tumor cell proliferations. They can travel to distant sites to promote the generation of pre-metastatic niches, and vascularization is augmented and endothelial and stromal cell differentiation is induced, leading to pro-tumor environments. And in immune responses, the immune responses can become deregulated in a manner that impedes tumor recognition and anti-tumor immune functions, with cytotoxic T cells induced to apoptosis, while NK cell proliferation is impaired and T cells differentiate toward a T regulatory cell phenotype. And in bone marrow, derived cells can be recruited to the tumor and pre-tumor tissue, where they contribute to cancer development. And so, as we can see, they're involved in a myriad of different physiological and pathological functions. And so that makes them a very interesting entities to study, but also because they are available in the circulation, and these entities can be interrogated and studied. They are also powerful tools for research and powerful for drug development, we think, and also for molecular diagnostics. So we think it's a very fertile environment at the moment, and I think this is because there's just been a confluence of key technologies and developments that have that have come along. And so, perhaps 10 years ago it was a bit more difficult to foresee that they would have such a huge impact. But now that these developments are coming forth, I think it's much easier to predict that they will have a transforming effect on research and on treating of human diseases. So the fertile environment, and I think, is very synergistic with EVs, in particular for precision medicine. And basically, just a quick summary, I'm sure everybody's aware of this, but it's our move from to a molecular basis for disease, and the identification of molecular and gene errors, and then the development of molecular interventions to correct them. So leading to customized and individualized treatment, and this is of course, represented by the dozens of targeted therapeutics and companion diagnostics that have been developed for cancer and other diseases. And although there has been huge advances, most diseases and most conditions are not yet treated with targeted therapeutics. But I won't list, as you can see, these are the commonly and well-known targeted therapeutics on the market approved now, but about 42% is estimated of the current drugs in development are now targeted therapeutics, and that number is expected to increase, as is the absolute number of drugs that will be approved in the future. So it could be hundreds of drugs in the next 10, 20 years. And also important developments in next generation sequencing and informatics and in other omic style technologies is very important. So sequencing of the human genome, of course, has propelled this whole field and area. Nowadays, the cost of exome and whole genome sequencing has dropped quite a bit, and the throughput is higher, and the bioinformatics available and data mining capabilities are moving very quickly. And of course, other omic technologies are developing quickly too, and may catch up soon. And these are all applicable to, of course, precision medicine, but in particular, in our interest is in analyzing exosomes, whether it's for research or for molecular diagnostics. And so among these other technologies are transcriptomics, proteomics, lipidomics, and metabolomics. And so these, as we mentioned earlier, since the exosomes are carriers of a lot of these molecules that would be amenable to this type of analysis, it's very exciting to see this technology developing and becoming accessible in terms of the costs, and also improvements in their sensitivity. There's also renewed interest in the ultimate, what I believe is the ultimate in personalized medicine, which is the use of the immune system to do the work for us. And so neoantigens are hot, and there's applications for exosomes in that field in many ways. And so, as we all probably agree, that disease is really a dynamic and relational set of events, and it's constantly evolving. I think that again, extracellular vesicles and exosomes are ideal for these developments and for combination with these other technologies. So here's just a representation of the various actionable genetic changes that have been identified, and then the drugs that are available for those to treat those. And again, that repertoire is growing quickly and will be very large. And so I think exosomes can one, help to monitor for disease, monitor patients, but also to help identify new markers, and even therapeutics, as we'll see as we move along. So EV and exosomes are uniquely positioned to catalyze change, and in particular for disease and therapeutic response monitoring. And we think that they will allow us to move from extrapolation to individualized approach, and so further the move to personalized medicine, and move us from an invasive to minimum invasive techniques and processes, and from a static to a dynamic approach, and as well as from single to relational holistic analysis of the various factors. And of course, all this will help us to be more proactive instead of reactive by being able to monitor even before disease and during disease and during treatment. And as the technology becomes more precise and is scaled down, miniaturized, as well as costs are reduced, there are already plans to develop point-of-care units that can do exome and full sequencing analysis. And so in the next 10, 20 years, I think this will be commonplace. And so now we're going to move into specific applications, and I think where the value can really be gained from this technology is in the research and development continuum. Excuse me. And here, what we're going to do is we're going to cover a bit of a case study, which is Alnylam Pharmaceuticals, as an example of what can be done with this technology. And so basically, we think that exosomes and vesicles would make an incredible, and actually they have been used as a platform for pharmaceutical and molecular diagnostic development. And I think they can support every phase of that development, including basic research, biomarker discovery, screening and targeting new targets, preclinical efficacy, absolutely, and preclinical safety. Also in clinical safety and toxicology to monitor clinical response. There's a lot of work being done now in translational and research and development of biosignatures. But beyond being used as tools, they can actually be used to develop new drug delivery systems and gene delivery systems and identify new therapeutic targets and actually function as therapies and as imaging agents. So moving on to the case study. So Alnylam Pharmaceuticals, they are a pharmaceutical company which is developing gene modulation pharmaceuticals. And their focus is basically in small interfering RNA, and they have a delivery system, which we won't get into here, so our interest is in the exosome use. But they also, in their development programs, study microRNA, antisense oligos, and viral vectors as methods of delivering therapeutics. And so this is very nice because they've used these various modalities using exosomes. And so it's a very strong and robust proof of principle that we'll see here. And so in doing this research, it requires quite a bit of monitoring of tissue-specific gene modulation, which means that in vivo, you essentially have to sacrifice the animals and excise the tissues and run various staining and processes to do the analysis for gene modulation. And so it's difficult, it's time-consuming, and it's very costly. And of course, in humans, if not impossible, it would be very limiting. And so the ideal monitoring system would be one that is non-invasive and temporal, that you could use across various stages of development, and ideally with the same animal. You don't want to have different cohorts of animals to evaluate different time points. So having the same animal with its own control is also a huge advantage, which of course is also an application in humans. And of course, you would want to see close correlation with tissue gene expression, and you would want it to translate against or across different models of disease, different species, and into the clinic with humans. And so I'm happy to say that they certainly have shown this, that it's a very good platform. And they, just to go back a little bit, they used a sort of a standard method of isolating exosomes. They used serum from animals, but also from patients, and they filtered and then used a low spin to remove the cells and debris. Then they used ultrasonification for a few hours, and then they extracted the RNA and analyzed their target via qPCR. And so in their case, most of the research prior to using exosomes required tissue and I'm sure they use it in parallel, but yeah, it required a lot more animals, a lot more work, a lot more expense. Now, in some of their studies, they have conditions where they can follow the condition using circulating secreted proteins, but in cases where those proteins are not available, the value of exosomes would be, and something where you could track gene expression from peripheral blood or other fluids would be tremendous. And so they've shown that that they could follow their targets in various bodily fluids using mRNA as their source material. And so they've shown that exosomes can be used to monitor RNA mechanisms in the circulation. And again, they mentioned that it would be valuable where proteins aren't available, circulating proteins. And so here's the one of many data sets that they show, and I recommend that you look up this poster. Many of the people I've spoken to are not aware of this. So anyway, they showed very close correlation, and there's a couple of experiments here that they run, A and B, and they ran the experiments, a time course in rat liver tissues versus exosome-derived mRNA. And so they treated these rats using a TTR silencing LNP siRNA therapeutic, and they injected the animals IV. And you can see the close correlation. The black is the tissue, and the gray are the serum levels of expression. And the first experiment, this is done over a period of days, from day 1 to day 14. And they basically are looking at the recovery of the downregulated genes. Whereas in experiment B, they're looking at an earlier time point, so they're looking at hours, and they're looking at the initial effect. And again, the correlation was very, very strong. Now that's in animal models, but they moved on to use the same technology in a phase 1 clinical study using this target, the transdirecting amyloidosis, or TTR for short, and it's a neurodegenerative disease, and it affects the liver. And amyloidoid deposits are really the pathology that is fatal to patients that have this condition. And so they used a gene silencing RNA therapeutic to target the tissues in the liver, and they monitored this liver downregulation using exosomes. And so they used TTR mRNA, the cleavage product, as their marker. And there's potential to use this technology for therapeutic activity monitoring, as well as potential to customize dose, maybe even for safety evaluations. And so this is published in the New England Journal of Medicine, and as their VP of Research states, they use this as a critical point in their drug development process. And here's reported data on their use in the phase 1 study, where our interest, they showed very good data, very good effects, which is great. But our interest is at the fact that they use the RNAi mechanism to confirm mechanism of action. They used analysis of circulating mRNA. They also use this in another study, so that shows that it's, again, very robust. And they used it, interestingly, both in serum and in urine, and they saw very similar results to the other previous phase 1 study. And it also reflects the data that they've shown, all the data that they generated in vivo, with in vivo models. So in conclusion, I think that it's a very robust platform that translates well across various species and across therapeutic time course, and also across the various therapeutic modalities that they use. So they evaluated this in vitro in mouse, rat, non-human primates, and human volunteers, and they showed very strong correlations there. They did it over hours, and they also studied it over a period of days, up to I think it was 50 days. And then they used it using various therapeutic modalities like siRNA, mRNA, antisense, and gene viral gene therapies, and they saw very good results. So I think this is a very good argument that this technology it worked well for them, but it has the potential to work for almost any therapeutic development program. And there's big interest from us in that, and we are working on various projects that are taking that into account. But beyond the research and development phases, they have applications across the whole disease and treatment continuums. And so, at least in monitoring and in diagnosis, they can be used, and publications have shown that they can be used to show predisposition for conditions, as well as in screening, diagnosis, prognosis, as well as monitoring for treatment, and also monitoring for drug resistance and relapse. Beyond that, they also there are programs to develop them as diagnostic imaging reagents. And as I mentioned earlier, they can also be used as to identify new therapeutic targets, or to use them as targeted drug delivery vehicles, and also in immunotherapy as sort of tumor vaccine type strategies. So now what I'm going to do is I'm going to move into our sort of seminal experiments and our early work as an example of molecular diagnostic applications. And so the premise here is that tissue is the issue, and I borrowed this from another presentation. And I'm sorry if I don't recall who it was, but I'll give you credit on the next time around. But anyway, tissue is absolutely the gold standard, and it's how we diagnose most conditions, including cancer. But it does have many limitations. Among those are limited sample availability, that the biopsy or tissue that is obtained typically represents a single point in time. It also represents a small region of the lesion, unless it's a full surgical resection, and that the tissue you may have representation. Even within a tumor, it's been found that different regions of the tumors have different characteristics. So your representation of that tumor may be limited. The other thing is that if you're looking at invasive cancer, you may not be representing those areas where the tumor has migrated, or even in metastasis, which is of course the most important for survival issues. But it's also invasive as a procedure, and it can be risky for the patient. It's expensive, and there's no possibility for continuous monitoring, and there's no possibility for individualized baselines or biosignatures using tissue. Whereas with a liquid biopsy style, exosomes, extracellular vesicles, you could potentially use them to begin monitoring individuals way before disease even presents itself. And to me, that would be sort of the dream project, to take 1,000 or 10,000 individuals and start monitoring them now, and begin to create that data set and to see where the disease arises and what are the triggers to that. We'll talk a little bit more about that in a bit. So anyway, the 2004, our hypothesis was that we could use the extracellular vesicles to move, again, this is a little bit of a recap, but extrapolation to an individualized approach, from an invasive to a non-invasive approach, from a static to a dynamic approach, and from a single to relational holistic analysis. And of course, all that would potentiate being more proactive rather than reactive. And as the costs are reduced to move from lab to point of care systems. And so this is a real paradigm change that's underway. And it's moving us from what I like to say is a static picture situation to a moving picture situation. So the traditional tissue approach, and we're using HER2 as a model because we, in our early work, so as I mentioned before, we were the first to discover the association of RNA and DNA and MVs, and so we selected HER2 as a model basically because it's very well characterized, and at the time there was already a therapeutic and a companion diagnostic, and there was quite a bit of research that was being done in this particular field. So I thought it would be a great model to test our hypothesis on. So in tissue, again, this is in tissue. The standard is to do immunohistochemistry for cell surface receptors, and when there's a question as to whether the results are affirmative or not, then they move on to FISH, which is a gene copy analysis by fluorescence in situ hybridization. Interestingly, there's plenty of work also in publications on PCR of this particular HER2 mRNA. And there's also the protein is soluble, so it can be found in the extracellular space. And so there was an ELISA developed for that, although I don't think it was developed into a clinical test that was at least successful commercially. But anyway, so it creates a very nice platform to use for benchmarking. And so a little bit more just background on HER2, just so it puts the experiments in context. So HER2 is a human epidermal growth factor receptor 2 oncoprotein, and 20 to 30% of all breast cancer cases are positive for HER2, with it being the second poorest prognosis for patients. So it's a very serious condition. Herceptin was a humanized anti-HER2 monoclonal antibody that was developed. By the way, there's a book called, I believe it's called HER2, which is very good. It goes into the whole development history on this. It's very interesting. Anyway, the patients, there was a companion diagnostic that was developed in conjunction, and so you have to be positive for HER2 under that companion diagnostic in order to be eligible for treatment. Unfortunately, only about 30% of those patients that are eligible respond, and the average extension on life is only about 9 months. And this is on monotherapy. I'm sure there have been improvements since then, and there are new therapies that target other variants of HER2. But also there's acquired resistance, which is a huge problem, and it's more and more recognized. And again, those new therapeutics and other combination therapies are addressing that, but there's still a huge opportunity for new technologies that can identify new markers and new methods of monitoring the patients for resistance and trying to capture it as early as possible. There's also the fact that some of these tumors are evolving, and so being able to identify other potential targets for therapeutics is very important. And so that's where the whole profiling, tumor profiling comes in, and ideally it being done in the liquid form would be best. Here's how you score the tumors, and this is important because they use these standard cell lines, SKOV3 as a high expressing HER2 a clone, MD361, I believe. Sorry, I can't see it too well, but anyway, and T47D and MCF7. So that's the gold standard, MHC, immunohistochemistry, and here's the micrographs and the different levels. And so again, a score of 2 or a score of 1 is considered equivocal, and so it has to go to immunofluorescence. And importantly, again, these cell lines are used as controls in actual clinical testing. So they are very important for our study design. And this is FISH, which is the fluorescence in situ hybridization, where they actually just do a gene count. And so if you get a gene count that's higher than 2, then you're considered positive. And if it's less than 2, you're considered negative. And therefore you qualify for treatment on that. So here's our first experiment, and this is very early on. But again, what we did is we used those same cell lines. We used in vitro, and the first chart here on the left shows the expression levels of the different lines in cells themselves. And so this is an expected result where the SKOV is quite elevated compared to the other two, which are somewhat negative. And so we did the same experiment, but using the media, the cell media, and the exosomes extracted from that. And so we could see that the results are very similar and correlate. And then we also did some agarose gel electrophoresis to confirm those results. And you can see here that the expressions are there, and also relative. So the SK over 3 expresses it quite high compared to the other lines. So of course, being in preclinical research, I know that a lot of times what you see in vitro doesn't translate to in vivo. So we moved on to a pilot study using xenograft models, and these basically used the high-level expressing SKLE3 cells. So we injected these in athymic nude mice, and we let the tumor grow till it was palpable, and then we took mice that were not bearing tumors, and we tested the serum from those mice. And again, we saw that the expression was very high in those that were carrying the tumor, and the ones that weren't did not express the tumor. And by the way, these are human tumors. So these are human tumors being expressed in mice. So there was a very good indication that that this could work in human serum. And so our very next experiment was in using clinical samples. And so we obtained frozen samples from biobank, and we obtained samples that had been identified as positive by immunohistochemistry. And so these were known positive cases. And we have here, compared to normal controls, so individuals without cancer at all. And so you see that there's definitely a stark difference between the two groups using PCR. The groups that were positive, there were of note. There was the number 5 is a stage 2, and number 4 and 6 are stage 1. So that was an early indication that you could identify the disease very early on. And so the advantages, and we think this is a potential game changer, is because the analysis of exosomes beyond, using a single marker like we did in those experiments, can be multifactorial. So you could look at various RNAs, not a single one. And indeed, most of the work now is looking at biosignatures of multiple mRNAs or mRNAs. Also multifactorial in the sense that there are different categories of markers: RNA, DNA, lipids, and protein markers. And so all of these can be used together, the proteins, to isolate subpopulations. We could look for gene alterations in DNA or gene expression using RNA, protein expression. We can also identify the cell of origin based on cell markers, or even the markers that are associated with cancer, and isolate those. So there's a potential here for very thorough relational analysis using transcriptomics, proteomics, genomics, and metabolomics. So as the cost is reduced, these can be used now for research, but as the cost is reduced, you can see that this potentially could be used even in a clinical setting one day. So again, to use the phrase again, so we would be moving from a static snapshot to moving pictures. And so going to a minimally invasive approach using biofluids, a dynamic approach, because we would be able to do limitless sampling and real-time monitoring, as well as a more personalized approach, since we would be looking at biomarkers and biosignatures for that specific individual. Also personal in the sense that we could establish baselines for an individual. And then it's relational. We could, using various omic technologies, and potentially, we could follow the individual full life cycle from conception to geriatrics, and that would be ideal. So I think that's what the future that we're looking at. Synergistic with existing and emerging technologies. And so, in conclusion, so what is the future, and what are some of the future challenges? And actually, this is inspired by a talk that I heard by Mustafa Rogi who's the Chief Technology Officer at Illumina, and he was talking about how complexity increases with disease progression. And I fully agree, and this is my representation of that. And so as we move from a hemostasis sort of standard in health to certain critical events that we may or may not be aware of that then lead to complex network dysregulation as we move further into disease. And so right now I see it as, the far right, where we're identifying dysregulation or markers that are already in a complex disease sort of environment. Whereas I think the ideal situation would be to sort of put the film on rewind and go find the points, the critical events at which dysregulation really started. And I think that's to me that's the holy grail, and I hope that that's where we're moving to in the future. So so we don't currently understand these critical events. What, where, and when do the driver alterations occur? So we know that these are time-dependent events, and they're patient-specific and they're relational. And so ideally, we'd like to. So the question is, do we do monitoring of genomic alterations or gene expression? And I think we have to do both, and we have to do it early, and we have to do it across the life cycle. So EVs and exosomes may be the key to have, because they have all the characteristics necessary to address this challenge. And so other active frontiers are the application of EVs and exosomes, and this could be for other talks, and there are other groups that are working very deeply in this as we are with various collaborators. But definitely in biomarker discovery and therapeutic target discovery, targeted drug delivery, immunotherapy, and in regenerative medicine. And to end the talk here, these are just a few of the companies that are delving into this field, either just looking to see what's there, or actually many of them have advanced clinical development projects that are underway, with some of them being scheduled to be commercialized in the next, maybe by the end of this year or early next year. So anyway, thank you, and now we'll move on to any questions. So there's a question here about preeclampsia and microRNAs, and I know that there's work already in that field. So, and I'm not privy to the data there, but I would encourage you to look those up. So, but yeah, there's definitely work being done there by other groups.
Thank you, Ray, for bringing that important presentation to us. That is just about all the time we have for questions. I know you did read that question that was in there. It was very long and complex, but we will definitely follow up if we didn't have time to get to your question today. We want to thank everybody for logging in, and just pass it back to Ray for any final comments that he has for our audience.
In its nascent stages, and so I look forward to being involved and engaged with all of you in the future. And thanks again. And thanks again, Brenda, for hosting.
You're very welcome, Ray. It was good information we had here from you today. I just want to let our audience know that this presentation will be on demand, available on our website through October 2016. You'll be receiving an email from LabRoots letting you know when that webcast is available for replay, and we would invite you to forward that email to any colleagues who may have missed today's live presentation. That's all for now. We hope we see you again here at LabRoots. Thank you so much, and goodbye.